Every one of you listen to me very carefully, friends, especially when you are answering the questions. Two things are very important, because you also know very well, friends, whatever you have done, whatever the subject that you have prepared in the 6 months or 1 year or 9 months, entire preparation can be, decided in 3 hours of exam. So that's why while you are analyzing the questions, you should be very calm, very patient, and very intelligent, especially then only you can answer every single question correctly. So make sure that you understand the question very correctly. Never get excited, don't think very fast, okay, because all the statements are very tricky statements. That's why have patience, analyze the question perfectly, then only give the answer. Otherwise, don't give the answer at all, okay. So now let's start the first shift, that is the part B as well as the part C. Again and again, I'm telling to all of you, I don't want your fast answers at all. I want an accurate answer. So make sure that you will read the question very correctly and you give the answer also very correctly. Take your time, not a problem. Okay. The question number 23. Take your time, no problem at all. Question number 23. Consider you're writing a final exam. Which one of the following mRNA is most likely to be exported out of the nucleus? Such a simple, straightforward question it is. Means what kind of mRNA can go out of the nucleus? That is the question. Option 1 says that spliced RNA associated with poly A binding and cap binding complex. Misspliced RNA with multiple stop codons for degradation in the cytosol. Spliced RNA with the associated spliceosomal complex, then uncapped and unspliced RNA. That is the question. Okay, everyone give the answers. Every one of you, every one of you. So, friends, we all people know very well, in case of a eukaryotic organism, whenever the mRNA got synthesized, friends, the mRNA is said to be the biologically active form of the mRNA. Sorry, it is inactive mRNA. We have to convert them into biologically active. If we want to convert them into biologically active, we all people know very well, we have to do 3 important steps. And where these 3 important steps are happening, undoubtedly, friends, these 3 important steps are happening within the nucleus of the eukaryotic organism only. Capping, splicing, then, friends, followed by the polyadenylation process. When these 3 steps occur successfully, then the mRNA become biologically active. It is ready to synthesize the protein. Hence, it can easily start importing from the nucleus into the cytoplasm. So what will be the correct answer now? The answer 1. Spliced RNA means the RNA which undergone the splicing with polyatyl and with the cap, that will only enter. They said that misspliced mRNA never enter. Spliced RNA with associated spliceosome. We don't require spliceosome first of all. And if the mRNA got uncapped, friends, it never ever enter into the cytoplasm. Hence, the correct answer is number 1. Simple question it is. Take your time, no problem at all. But I want you to give me the correct answers. Question number 42. Which one of the following translation factor is used at the step of translation initiation and defined as an anti-association factor for the 30S and 50S subunit interactions? Every student has to understand one simple point, that is called anti-association factor. This is more important that you people has to remember compulsory. They're asking anti-association. See, when I use the word anti-association means what, friends? This is the small ribosomal subunit and this will be the large ribosomal subunit. If every student quickly recall, in case of a prokaryotic organism, IF3 and IF1, these two associated with this small ribosomal subunit. And what I told to all of you friends, whenever IF3 is present, IF3 never ever allow the large ribosomal subunit to associate with the small ribosomal subunit. Then undoubtedly, what could be the answer? IF3, which is acting as an anti-association. IF1 do not have any anti-association. IF2 with the help of GTP, friends, we all know very well it is going to bring the tRNA molecule. Ribosome releasing factor or ribosome recycling factor, it is responsible for what? I mean to say, disassembly of the mRNA, tRNA, rRNA, all. Disassembly can be mediated by the RRF. But they are asking how the 50S and 30S can be dissociated. I mean to say, they never associate with each other with the help of IF3. Hence, the correct answer will be the 3. Very good. So, see here, in the morning shift, we got 2 part B questions. Now, this will be the 3rd part B question. Which one of the following codon is used to code for the selenocysteine in the Escherichia coli? Take your time, don't be hurry. Slowly, slowly, I'm telling you, all of you slowly. I don't know why you are so fast. I don't know when people understand my words. See, friends, it's not important whether you're intelligent or not. It is important whether you're having patience or not. Always remember my words. Don't be hurry. What happens if you wait for 20 seconds? What happens if you wait for 30 seconds? Nothing will happen, no? Okay. Others, everyone give me the answer. Swati said 4. Ruchi said UG. Tell me, Ruchi, what is the option? All the students give the option. Padmavati, Nitya, everyone. Santosh, what is the option? Ritika, Aishwarya, what is the answer? I am requesting you, don't sit silently, okay? I want you people to answer the question correctly. I don't know. See, this is the question of an intermediate level, which is a plus 12 level, right? What is the question they are asking? I don't know what you are looking at the question. Which one of the following codons is used to code for selenocysteine in the Escherichia coli? This is the question. See, friends, we have started studying the selenocysteine is coded by a stop codon, UGA. Always remember this. And I want you people to remember this also. Some people has given the answer as the 3rd one. Pyrrolysine is the amino acid which is coded by the UAG. Remember, pyrrolysine is coded by the UAG, friends, and the selenocysteine is coded by the UGA. Now you have to ask me a good question. Sir, UAG and UGA both are the stop codons. Then, sir, why you are saying that these two are coding for this amino acids? All the students remember, when we are reading the translation, I have told you one important statement. I don't know whether you remember or not. Amino acids are present everywhere. Means, friends, amino acids are present in every organism. What, friends? Amino acids are present in the every organism, but not universal. In other words, they are widespread. I hope you are following my statements correctly. They are widespread. What do you mean by the word widespread? They present everywhere. They present everywhere. But friends, they are not same in every single organism. What I mean to say, UAG, UGA present in every single organism. Try to understand me correctly. UAG and UGA present in every single organism. But the point is that in every single organism, they are not going to code for the stop codons. That is called as amino acids are widespread, present everywhere, but they are not universal. Selenocysteine is coded by UGA. Remember, pyrrolysine is coded by the UAG. Hence, the correct answer will be the number 1. 70% of the people has given the a wrong answer. Don't get confused with this particular point. I want you to understand slowly, analyze it thoroughly, and then only give the answer. Okay? No problem, no problem. Rectify your mistakes. Rectifying mistakes is also very important, friends. So we have solved a 23 question, 42 question, 45 question. So in the part B, how many questions we got? 3 questions we got. 3 into 2, 6 marks. Okay. And what is the luckiest part? The luckiest part is that each question is a very, very easy question. So you should not miss this particular questions. This also one, some people answer one question wrongly. Slowly think it. Don't be hurry. Now, these are all the 4 marker questions. I'm requesting all of you, please be very slow. Take your time of more than 1 minute. Take more than 1 minute, no problem. But make sure that you'll understand the question correctly. The following statement refer to factors regulating the fidelity of the DNA replication. Every student commonly know, right, friends? What is fidelity, friends? Fidelity means accuracy of the DNA replication. So to maintain the accuracy of the DNA replication, what the DNA polymerase or what the enzymes are going to do? That is the simple question they are asking. Take your time, think it correctly and answer. First, option A says that 5' to 3' exonuclease activity of the replicative DNA polymerase, imbalanced intracellular concentration of 4 DNTPs, increased intracellular concentration of RNTPs resulting in increased incorporation of RNTPs during DNA synthesis which are not readily removed by the polymerase proofreading activity. Option D, removal of incorrectly incorporated nucleotides by mismatch repair system. And I want all the students has to try your best in eliminating the option. Use your brain in elimination. Always use elimination. This is the part where you have to use your intelligence. Okay, you take your time, but give me the answer. I'll explain you properly. Sisira, Santosh, whatever the answers you people has given, you recheck whether you are confident or not. Ruchi said 2. Ritika 2. Others. Every one of you. Ranjita, what is the answer? Padmavati, what is the answer? Aishwarya said 1. Okay. Mahesh, Karan, quick. Okay, take time. Yeah, okay. What about others? Oh my God. Okay. Okay. Now every Padmavati answer my question. Every student answer my question. The following statements refers to factors regulating the fidelity of the DNA replication. So among the 3 type of polymerases, Pol1, Pol2, Pol3, which is the major polymerase? All the students answer my question. Which is the major polymerase enzyme? Every student answer my question, which is the major polymerase enzyme? Yes, you all people has given answer as polymerase 3. And every one of you answer my question. The polymerase 3 enzyme will do the proofreading in which direction? My question is very clear. The polymerase enzyme will do the proofreading in which direction? All the students has to answer. Then Padmavati, how it could be A will be the correct answer, dear? Huh? If you eliminate this option also, you will end up choosing the right answer. The 5' to 3' exonuclease activity of the replicative DNA polymerase, that is wrong. Replicative DNA polymerase is obviously polymerase 3. Undoubtedly what? It will do the proofreading only in which direction? 3' to 5' direction only. Yes or no? Of course, yes. Right. So option A is wrong. If you come to know that option A is wrong, you can eliminate option 1. You can eliminate option 4 also. 80% of the students has given as option 1. And you have 2 options left out. Let's see, option 2 and 3. Now, no need to get confused. They're asking, friends, how can we maintain the fidelity or how can we maintain the accuracy? That is the question. imbalanced intracellular concentrations of the 4 DNTPs. What, friends? Imbalanced intracellular concentration of the 4 DNTPs. Yes or no, friends? Suppose example, if adenine, guanine, thymine are more in percentage. Suppose cytosine is in a less percentage. How does the DNA replication occur accurately? Tell me. How does the DNA replication occur accurately? It never occur accurately, right? So undoubtedly B will be the correct option. Suppose most of the students are getting confused between the C statement. So if you don't know about the C statement, you can come back to the D statements. You can easily understood. Removal of the incorrectly incorporated nucleotides by the mismatch repair system. Yes or no? You studied in the BSc, you studied in the MSc, and we are going to read in the DNA repair mechanisms also. Correct or not? Whatever the nucleotides that are wrong, by using a mismatch repair system, we will remove that and we will repair them. So B is correct, D is also correct. Then only you can come to the answer as option 2. So how you can answer this with the patience? At least you have to spend 1 minute. Automatically you'll find the answer. Option C says that increased intracellular concentration of RNTPs, ribonucleotide triphosphate, resulting in increased incorporation of RNTPs during DNA synthesis, which are not easily removed by the polymerase proofreading activity. Of course, true, friends. See, DNA require what? DNTPs, right? Right. And we only add the primer, which is the RNA primer, which are the ribonucleotides. But if you keep on adding the ribonucleotides only during the process of the DNA replication, it is very difficult. It is very difficult for the DNA polymerase enzyme to remove them. Obviously, true statement it is. Hence, B, C, D, all are correct. Hence, the correct answer is 2. Okay, quickly tell me, is it clear? Everyone understand this question or not? Everyone give the reply. That is not a problem. You can answer again once you read 2 to 3 times. It's easy. It's easy. Okay. Now, question number, what was that? 19. See, friends, I have requested every one of you, please read the antibiotics. I strongly believe that everyone read the antibiotics. Okay, it's a 4-marker question. Which one of the following statements is correct in the context of erythromycin-mediated inhibition of the protein synthesis in bacteria? I have explained you many number of times in a given slide. In the PPT also, it was there. Any one of you, please tell me. Ritika, tell me what is the mechanism of action of the erythromycin. I want any one of you give me the answer. Any one. Or at least I am asking all of you, erythromycin will come and attaches to the which site? Block the exit of the peptide. Okay. Anyone else? Yeah, it is there in the slide itself, na. What about others? Which indicates that no one is reading. Okay, you're just listening the lesson and leaving the concepts. So this is not the way to crack the CSIR exam. Remember, friends, if you don't read, there's no point. Anyone else can give me the answers? Okay, no problem at all. Right or wrong, everyone of you give me the answer. Okay. Okay. Not a problem. Not a problem. Okay. All the students look into this point. All the students look into the point. What was written here? Erythromycin inhibits translocation by the prokaryotic large ribosomal subunit. This is the question they have asked. What do you mean by the word translocation? Which is occurring when the peptide bond got formed. Means after the elongation, the erythromycin is blocking the translocation, which means movement of the ribosome from one codon to the another codon. Now all the students answer my question. Every student answer. My question is very clear to all of you. The erythromycin amino acid blocking the step of the elongation, right? Which means that, anyway, let me explain you briefly. This is the E site, P site, A site. So here first tRNA comes and attaches. Next tRNA will come and attaches here. Now what happened, friends? The first peptide bond got formed over. Then the tRNA from the P site, it will come to the E site, correct na? Then the tRNA present at the T site with the A site with the two amino acid, it is coming to the P site. Now which site become free? A site become free. But if the ribosome, if the new tRNA has to come here, the ribosome has to move. Ribosome has to move. But entire translocation process blocked by the erythromycin, which means that the ribosome never ever move. No translocation occurred. As a result, the peptide bond synthesis, protein synthesis will stop immediately. Now, my question to all of you is very clear. First peptide bond formed or not? My question to all of you. First peptide bond formed or not? Every one of you. First peptide bond formed or not? Of course, yes, right. The first peptide bond got formed. It is only blocking from the elongation. You're getting my point. So that is what they have asked the question. Look into the given question. Which one of the following statements is correct in the context of erythromycin-mediated inhibition of the protein synthesis in bacteria? Listen. Erythromycin inhibit the protein synthesis at the step of the elongation. Every student commonly know that. But it nonetheless allow the translation of the first few codons, especially codon 1 and codon 2. The peptide bond got formed. Is it? The answer is number 1. Option 2 says that erythromycin inhibit the protein synthesis at step of elongation. This is right. And it prevents the formation of the first peptide bond. Wrong statement. Option 3 says that erythromycin inhibit the formation of a translation initiation complex. Wrong statement. Erythromycin is toxic to the bacteria because it results in the initiation of protein synthesis with the elongated tRNA. The statement 4 doesn't make any sense. They are saying that initiation can be started from the elongated tRNA. Means they are saying that initiation will start from the third or fourth tRNA. How is that possible? Initiation can be started only from the first tRNA, which is complementary to the AUG. Hence, the correct answer is number 1. Is it clear to everyone or not? Yes or no? This is a very common sense. It's not a common sense, nothing. It's a direct question. You people can answer it easily. But the point is that you have to know it slowly. Again and again and again, I am saying, friends, I don't mind to repeat these lines. It's not important how intelligent you are. It's only important how patience you are having. When you are having patience, you can analyze properly. That analysis will brings you the confidence. You can sort out the right option. Slowly think it. Not a problem, not a problem. Okay. So we have solved 2 part C questions. There are total 5 part C questions were given. Take your time. It is truly logical question. If any one of you attended what we can say that lac-operon, tryptophan operon live class also, you can answer the question. It's a purely logical. I'm sorry, it's not logical, statement related. But you have to know it. If you know it, you can answer. You take 2 minutes of time, no problem. Take 2 minutes of time. Read it. Every line is there in the PPT also. Every line I have explained you. I believe at least 2 to 3 people will give the right answer. The statements only makes us confused. But to be frank, the statements are logical. We have to find that logic with patience. You have to choose the correct statements. Oh my God, this much fast. Okay. But Nitya, you have studied last time, right? You try it. It's a pure conceptual only. Try it. You have the concept already. Tried it. Try it. Give it a try. Ruchi, try it. Think slowly. You can answer the question. It's easy, friends. If you see the lack operon, you can answer the question very easily. Okay. Okay, others. Everyone given answer as 4. B is wrong. Oh my God. Okay. Okay, others. It's okay. Right or wrong, give the answer. Not a problem. Give the answer. Give the answer. So C and D. Okay, others. Mahesh, what is the answer? Ranjita. Who's this Pawan Kumar? Okay, Pawan Kumar. I don't know who's this. Okay. Oh, okay, okay, Archana. Okay. Okay, Archana, try this question. What's the recorded without fail? Okay, who can give this answer? Okay, everyone given the answers. Ranjita said 1. Okay. Now let's try to understand the question. It is not at all difficult, friends. It's easy question. But the statements are very tricky statements, make you super confused. The lac I and TRIP-R repressor bind with their ligands allolactose and tryptophan respectively, resulting in alteration of their DNA binding properties, right? The following statements are made about the mechanism of lac-I inducer, lac-I regulatory gene, TRIP-R repressor, binding with DNA operator, and the regulation of gene expression in the E. coli. I will show you here itself. It is a direct, straightforward, simple question. Just give me 1 second of time. I'll show you right away. Okay. Now, friends, let's look into the state. 1 second. Just give me one moment of time. Let's look into the statement 1. What is the statement 1? All the students listen. Allolactose binding to the lac-I leads to its poor binding to the lac operator, whereas the tryptophan binding to the TRIP-R leads to its better binding to the tryptophan R to the TRIP operator. Yes or no? All the students look into the option A. I am explaining you point by point. Allolactose binding to the repressor protein. When allolactose bind to the repressor protein cannot attach to the operator. That is absolutely a true statement. You only look into this particular picture. All the students pay attention. You no need to get confused here. We all people already studied this. Just give me a moment. By that, you never forget where I show to all of you. You only look into the picture. All the students look into this beautiful picture. You are getting a very clear-cut idea. See, friends, you can see this, right? What is this? This is the allolactose. All the students listen. allolactose will come and attaches to the repressor. When the allolactose will come and attaches to the repressor, do you think repressor will go and attaches to the regulatory to the operator? Never ever. So friends, option A, in the option A, the first statement is absolutely true statement. It is a right statement. Now let's see the second statement. In the tryptophan binding to the tryptophan R to the repressor, every student remember friends, in case of a tryptophan operon, repressor is inactive. What? Repressor is inactive. When the repressor become active, when the tryptophan will come and attaches, then the repressor will become active. Then the repressor will go and bind to the tryptophan operator very tightly. So see here, that is only we have written here. All the students, please look into the diagram. You automatically understand this. Every student look into this picture. Every one of you. This is the repressor. When the repressor becoming active, whenever there is a tryptophan comes and attaches. So friends, what's the function of a tryptophan? Tryptophan can act like a co-repressor. So in the presence of co-repressor, repressor become active. Repressor will come and attaches to the operator. That is what they have given the option A. So listen, you please ask me if anyone having any kind of a confusion again and again. Yellow lactose binding to the lac I leads to poor binding of the lac operator. Why? Because yellow lactose block the repressor. Then the repressor cannot attach to the operator. Right statement. But tryptophan, when go and attaches to the tryptophan repressor will be super active. The moment it becomes super active, it will go and block the operator. Option A is absolutely right option. If you come to know that option A is a right option, you can eliminate option 3. You can eliminate the option 2 also. Now let's look into the option B. Now you tell me, yellow lactose, what is yellow lactose? Inducer. Where it will bind to? It will bind to repressor. By that, the repressor will block, which we can call what? See friends, you tell me, if the repressor got blocked, every student has to answer my question. When the yellow lactose will come and block the repressor, the operon will be turned on or turned off. Every student. The operon will be turned on or turned off. All of you. Everyone. Undoubtedly turned on. That is only they are saying that yellow lactose binding to the lac I leads to the induction of the lac operon. Right statement. Now you people answer my question. When the tryptophan will come and attaches to the tryptophan repressor, operon will be turned on or operon will be turned off. In case of a tryptophan operon will be turned off. That is what they are saying in the option B. So option A is correct option. Option B is correct option. Hence the answer is number 1. Option 3, option C says that binding of yellow lactose and tryptophan to the lacite trip R both will cause the repression. Option C is wrong, right? How it will cause both repression? Yellow lactose will induce the operon, tryptophan repressor will block the operon. But they said that both are blocking the operon. Undoubtedly, option C is wrong option. Now, when you look into the option, anyway, you got the answer already, option A and B. Now, option D, binding of yellow lactose and tryptophan to lac-I and trypt R respectively, okay, leads to activation of their corresponding operons, right? But this is also wrong statement, right? Only yellow lactose will cause the activation of the operon. Trip R repress the operon. That statement also wrong statement. However, in the tryptophan operon regulation, availability of tryptophan also result in the attenuation mediated transcriptional termination, leading to an overall effect of the repression of the tryptophan operon. Why this statement is wrong? You all people commonly know that we have a 2 gene regulation, negative gene regulation, positive gene regulation. Positive gene regulation is what? Attenuation. What is the function of attenuation? Attenuation function is what? Blocking the complete polypeptide synthesis. Correct, na? So this statement is a true statement. From here, this statement is true statement. But what is the upper statement? Binding of yellow lactose and tryptophan to the lac-I and R both causes activation. That is wrong, right? Only yellow lactose will turn on the operon. Trip R repress the operon. So hence the option 4 also wrong option. Hence what is the correct answer? The correct answer is number 1, A and B. Is it clear, everyone? Let me know. I will re-explain if anyone having any other difficulties. Any point that you did not understand, let me know. Is it clear? 90% of the students will make it wrong. What is the reason? Because we fail in reading the statement properly because of our excitement. Only that thing will happen. That's all, nothing else. Others got it? Ruchi, you got it correctly. Whomever given the wrong answers, try to understand the points correctly. Understand, Sisira, Swati, all people understand? Okay, got it, got it. Not a problem. Okay, great, great. Okay. Now the next part C question. Take your time. Oh, what's the question number? 65. Take your time and answer the question. Don't be hurry, okay? Archana, I will explain that. Because Archana, in case of attenuation, you know, right? In case of attenuation, see, whenever the tryptophan is not present, listen my words. Archana, listen. Whenever the tryptophan is absent in the medium, when the tryptophan is absent in the medium, already in our body, tryptophan is getting biosynthesized. Yes or no? See, suppose 40% of the tryptophan mRNA got biosynthesized. Is it? Now, all of a sudden, you have provided the tryptophan. When you provided the tryptophan, now I am a bacteria. I don't take the tryptophan. I will start taking the tryptophan whichever you have provided to me already. Now you tell me, what should I do now? In my body also already little amount of the tryptophan is already synthesized and it is going on. So I will block the elongation. I will block the termination also. Now the point is that already little amount of the tryptophan got synthesized. Tryptophan mRNA got synthesized. Very little part, very, very little part. Now the question is that, does this little part is biologically active? No, this little part is a biologically inactive. Why? Because it can produce a truncated protein. Truncated protein means the protein is not completely synthesized. Only initiation is over. When the initiation is over, I provided the tryptophan externally. We blocked the elongation and termination. Whatever the tryptophan biosynthesis occur, it gives rise to only a small protein. So we are blocking the elongation and we are blocking the termination by using this attenuator. So that is what we generally call it as a positive gene regulation by the attenuator mechanism. You got it correctly. Now, anyway, friends, this question is a very straightforward question that every student can answer it easily. Whenever nucleotides got break down, if I want to break down the nucleotides, what bonds I have to break? Phosphodiester bond. If I break the phosphodiester bond, nucleotides will be separated. Only nitrogenous base. We all people commonly know that, friends. Suppose this is the sugar. Sugar, with the help of a N-glycosidic linkase, it is associated with base. This is called N-glycosidic linkase. When I cut this particular N-glycosidic linkase, sugar and base is getting separated. So the only way of separating the base is cutting the glycosidic bond. So nitrogenous bases break down the glycosidic bond. A nucleotide contain a sugar, a nucleotide contain a base, a nucleotide contain a phosphate group. When I cut the phosphate group, I left with only sugar and base, which we together call it as a nucleoside. So which is the removes the phosphate group. This is one strand of DNA. This is another strand of DNA. All the bases connected with each other in the two strands of the DNA by hydrogen bonds. When I break down the hydrogen bonds, what I am going to get? I am going to get the single stranded DNA. So the correct answer will be the 2. Okay. No need to get confused, friends. You can answer this question very easily. Even though you have a small common sense, you can answer this. Question number 67 for marker. Take your time. I encourage all of you to eliminate the option. Trust me, if you eliminate the option, you can choose the answer just like that. Eliminate. I want you to eliminate. That you have to learn. It's okay. Upon practice, when you do the practice of the same question second times, it will become super easy. If you have a small knowledge on the telomeres also, you could answer the question. Okay, someone has given answer as 4. Okay, people has given option as 4. Okay. I want every student has to answer why the option B is wrong. I want every student has to answer why the option B is wrong. Ritika, why option B is wrong? No, no, no. You people think and answer the question. No problem at all. First you think it. Okay. Deepika said 2. Hmm. Anyone else? See, look into that. Answer is the 4. Okay. Look into that option A. Anyway, all of you listen. All of you listen. I understand your concern. Listen. Telomere binding proteins, we can call TBPs, are believed to shield telomeres from the cell's DNA repair machinery, preventing them from being recognized as a double-stranded breaks. I have told to all the students many number of times. Okay, first of all the students answer. We all studied while discussing the lesson. The telomere sequences or the telomerase enzyme plays a crucial role in the cancer biology. Is that statement is a true statement or a false statement? That is my first question. The telomeres plays a crucial role in the cancer biology. That is what I told you in the lesson. So undoubtedly, option D will be the correct option. So you can eliminate option A, option 1. You can eliminate option 2 also. This is how you have to do it when you don't know the options correctly. Now, option A. See, in 1 minute, I can find out the answer. Option A. So I have eliminated option, what we can say that. What option I have eliminated? I'm sorry. Yeah, I have eliminated these 2 options. Why? Because in these 2 options, D option was not there. Now, option A says that telomerase binding proteins are believed. We discussed that this is the telomere sequence. What is the function of a telomere? It is going to protect the DNA from the degradation, and it is also involved in increasing the length. So if I comes to know that option A is a right answer, within 1 minute I can choose the answer as the 4. Now, why the option B is wrong? Some people are getting the doubt. And see the option C first. In differentiated cells, telomerase is inactive, leading to shortening of telomeres over hundreds of cell divisions, damage to ends chromosome, and eventually apoptosis. Yes or no? See, in a highly differentiated cell, don't think that the telomere is inactive for the long period of time. It is inactive for a particular period of time. Because of that particular inactiveness at a particular period of time, the telomeres are becoming shortening, and shortening, which leads to what? Apoptosis. Because friends, once the ends are becoming shortening, aging will occur faster. Once the aging occur faster, what is happening? Aging occurs, the cell will die very faster, which we generally call it as apoptosis or the programmed cell death. Now you look into the option B. All the students listen. There is a considerable similarity in the simple sequence repeat. For example, TT, GG, GG. It is the sequence where, friends, in the paramecium. TA, GGG in the trypanosoma. Here what they have given, TTA, GGG. Look into this, TTA, GGG, where this particular sequence is present. One second. Yeah. So this particular sequence is present in the Arabidopsis thallion. You're getting my point. TTA, GGG, only till this sequence. TTA, GGG. This is the only sequence in case of a human beings, or the even I can't say that, friends, it is also purely human being. Okay, I can say that at the current generation compared to the Homo sapiens, the sequence also completely got changed. And suppose example, so in the Homo sapiens, this is the sequence. In the Homo sapiens, TTA, GGG is the sequence. I agree with you. Let's consider the sequence is also right. But the point is that it never extend up to 150 kb. Friends, you know what do you mean by the word 150 kb? 150 kb means you can get the nucleotide sequence of the 10 plasmids. 10 plasmid sequence will be more than 150 kb. Do you think one single telomere sequence can be extend up to 150 kb? It never be that much. See here how many nucleotides, hardly it is 50 to 300 nucleotides, that's all. But they are saying that it is extended up to 150 kb. 150 kb means, friends, nearly 1 lakh nucleotides will come. That statement is absolutely wrong. Here sequence is also wrong, or the sequence is 90% similar, but they never extend up to 150 kb. It is hardly 100 to 300 nucleotides, that's all. Hence the option B is the wrong statement. Is it clear, every one of you? Archana, you understand now? You read the explanation, you automatically get it. Understand, everyone? Perfect. No need to get confused with that particular point. Okay, so total how many questions were given in one single question paper of the morning shift? So we got question number 23, question number 42, question number 45. These two are the part B questions. Question number 15, 19, 42, 65, 67. How many are there? 1, 2, 3, 4, 5. 5 into 4, 20, 20 plus 6, 26 marks from one single chapter. Then imagine how important it is. And ecology, we got 34. Developmental biology, we got 30 marks. No chapter we should not skip. Okay. Anyway, no problem. Slowly think it. Okay, now let's go to the afternoon shift. Okay, right now we understand the morning shift. Okay, now let's go to the afternoon shift. No need to get confused. I'll give the question paper also, don't worry. Just think it. I will explain this to all the students. You people try it, use your brain and answer. Oh, okay. Analyze correctly. Everyone try this question. You can answer this. If any student know what is nonsense mutation, every student can answer the question. Any student. Because anyway, we read the nonsense mutations in the genetics chapter, but you read in the BSc, you read in the MSc, everything you read it. If at least you know what is nonsense mutation, you can choose the right answer. Okay. Padmavati, Ritika, tell me what is nonsense mutation? Archana, tell me, after the nonsense mutation, the protein synthesis will occur. That is my question. You all people read many number of times this is. After the nonsense mutation, the protein synthesis will be continued. That is my question. No. Nonsense mutation means the mutation will be completely stop the synthesis of other genes also. Suppose friends, first nucleotide, second nucleotide, third, fourth, fifth, sixth, seventh, eighth, ninth, 10th, 11th, 12th. Like this, many nucleotides are there. Where the nonsense mutation occur, example exactly here at the sixth codon, nonsense mutation occur. Say example, it leads because of the mutation, it leads to a stop codon called as UGA. Now my question to all of you, when the nonsense mutation occurred at the sixth codon, do you think 7, 8, 9, 10, 11, 12 codons can give rise to the protein? Never ever. It never give rise to any protein. Okay. Now you think that same an answer. You think it. It's the name, that's all. Oh, super fast. Others. Okay. No need to get confused. You have to know this name, that's all. In lac-operon, a nonsense mutation occurred in the beta-galactosidase. Okay. In the lac-operon, we all know that we have a lac-Z gene, we have a lac-Y gene, we have a lac-A gene. So how many number of times we all people studied in the concept? Friends, in case of a prokaryotic organism, it is a polycistronic, which means that, friends, the three enzymes can be transcribed as a single mRNA. That is what we all people studied many number of times. Now the question is that among these three, who can synthesize first? Lac-Z synthesize first, followed by lac-Y, followed by lac-A. Then three can come out as single mRNA. Where the mutation occur, lac-Z. Now, do you think this mRNA synthesis will be continued, every one of you? Do you think mRNA synthesis will be continued? No. Lac-Y is blocked, lac-A is also blocked. What do you call that mutation? That is the question. It is called polar effect of the mutation. Okay. When the nonsense mutation occurred at a particular gene, which is blocking the synthesis of the further genes, that mutation only, friends, we generally call it as the polar effect of the mutation. That's a right statement. So question number 6, what is that question number 19 or 12? What is this? 1 second. Yeah, question number 12. I have explained you in the 2 classes before only, in the post-translational modifications. I have also explained you in the eukaryotic gene regulation also I have explained you. Several proteins are modified by phosphorylation at specific amino acid residues to alter their activities. Which one of the following amino acid is not typically a site of the phosphorylation in proteins? So now my question to all of you is serine, threonine, and tyrosine. These three are having what? We all people know very well, friends. These three are commonly having a OH group. Correct, na? So this OH group undergo the phosphorylation by certain kinases. Now my question to all the students is very, very clear. My question to all of you, tell me Ritika, tell me Archana, tell me Swati. Recently I told you, right, what modifications can undergone by the amino acid lysine? Every student. Tell me Santosh, Sisira. 2 classes before only we studied. Very, very good. Very good, Aishwarya. Acetylation also wonderful. And methylation also. 2 modifications. Very, very good. I want you to remember both modifications. Don't remember only methylation or only acetylation. It undergoes both. Major modification only acetylation. Because of the acetylation only, right, histone proteins become weaker. Then followed by the methylation. Right? Very, very good. Anyway, the answer is lysine. Wonderful. Then question number 26. Let the plant physiology get over. You can answer little more easily. The only thing is that you have to read correctly. Okay. Direct question it is also. Introns in the eukaryotic GNAs are found in. Where the introns are found? Use your brain. This is called trapping question. Slowly. This much fast. Whomever giving this much fast, you are not reading. Okay, that's good. Even though you are reading, make sure that you analyze correctly. See, one common sense is that, friends, we all studied rRNA, we studied mRNA, we studied tRNA. All the three RNAs has to undergo the modification. Yes or no? That is common point. Everything has to undergo the modification. So as a part of the modification, undoubtedly, we are going to remove the introns. The answer will be the D. Now, they are just giving not in the tRNA, not in the rRNA, not in the rRNA and tRNA, just to make us trap, just to make us super confused. So that's why you should know the concept correctly. Now, you try to understand, is this kind of things are required a very depth of the concept? No, these things you have to remember. The moment you remember, you can answer this. Okay. So these are all the different type of RNAs which are present in the tRNAs, which are present in the rRNAs, which are present in the mRNAs also. What is this question number? 26. The question number 30. Oh, sorry. Think it, slowly think it. I want you to answer. It is also a simple question, makes you confused. In eukaryotic genes, DNA sequence that define the gene promoter occurs. Take your time, no problem. In eukaryotic genes, DNA sequences that define gene promoters occur. See, now all the students answer my question. In case of a, suppose this is the INR, initiation region. Correct na? My question to all the students is very clear. In case of a prokaryotic organism, the promoters are present in the upstream or downstream? All of you. Is it present in the upstream or downstream? Only it is the upstream. What about the eukaryotic organisms? We all studied in eukaryotic organism, it is present in the upstream also. It is present in the downstream also. So instead of using the word downstream, they used that within the transcribed region. Because friends, this entire region is undergoing the transcription. So under this region, only downstream promoter is present, right? So that's what they have given. Either in the regions upstream of the transcription start site or within the transcribed region. In the transcribed region, only downstream is present. Nothing is present at the termination site. Only in the regions that present the no. Only in the upstream, no. It present both upstream as well as within the transcribed region. Very good. Answer will be the D. Right. Okay. What's the question number 28? Okay. Think slowly, analyze it correctly and give the answer. Okay. This is the direct question which directly come from the slide itself. 4-marker question. How easy question it is. It's there directly in the slide itself. Okay. What happened to others? Yes, we all people clearly studied, right? Friends, the amino acyl tRNA synthetase is the enzyme which will add the amino acid either to the 3' hydroxyl group or to the 2' hydroxyl group positions. Yes or no? It's a straightforward question. But if you ask me a correct point, most specifically it added where? Most specifically, it added the 3' hydroxyl group. But what did I told to all of you? It may add at the 3' also, or it may add at the 2' also. See, friends, this question they have asked for 4 marker. How easy question it is. Okay. The correct answer is A. Good. What's the question number? This is 28. Okay, question number 33. Slowly think it, analyze it, and answer the question. No need to get confused. Simple question. I'm expecting everyone give the right answer. See, friends, from the translation only, they have given 2 4 markers. Understand that, how important it is. Following statements are made about the bacterial ribosomes and their functions. Take your time, no problem. Read it correctly. Easy, friends, easy it is. Seriously? Santosh, okay. Others, every one of you. Ritika said C, Deepika C, Santosh is B, Nitya C, Swati C, Pawan C. Others, Padmavati, what is your answer? Okay. Every one of you, first of all, answer my question. Every one of you. A ribosome carries the polymerization activity. Is this statement is a true statement or a false statement? Tell me, everyone. Is this statement is a true or false? Yes, it is a true statement. Let's eliminate the option A also. Let's eliminate the option D also. Now, my question to all the students is very clear. Where does the peptidyl transferase enzyme is present? Is it present in the large ribosomal subunit or is it present in the small ribosomal subunit? Nitya, what is this? You forgot everything, huh? Where it is present? All the students. Where it is present? Very, very good. It is present where, friends? In the large ribosomal subunit. 50S and 30S. 30S contains 16S. 50S contains 23S. Here the large ribosomal subunit. Here peptidyl transferase enzyme is present. Now my question to all of you. Here peptidyl transferase enzyme is present, which is involved in the peptide bond formation. What is the use of the 16S here? The option A says that, first you look into the option B. The 23 sRNA is necessary and sufficient to catalyze the in vitro peptide bond. My friends, they are not asking in the in vivo. They are asking in the in vitro. Undoubtedly 23S is sufficient. B is correct, C is correct. Option D says that the 16S rRNA is necessary and sufficient to catalyze in vitro. No. 16S rRNA don't have the enzyme. Why it will be used? Option A says that both are required. That is wrong. Answer is what? Option B and C. Correct answer is B. Is it clear, everyone? Understand it correctly. It's a simple question. Only 1 or 2 people has given the right answer. That's what. Slowly analyze. Don't be hurry. Once you, obviously it's a silly mistake. Once you choose the wrong option, you can't get it back. So that's what we have to do it very slowly. Okay. Question number 47. Friends, this is the question, is a mixed. Every single line we have discussed in the PPT, in the class, every single line we discussed. I want you to think, friends, correctly. And the question is correct statements. Oh my God. Who has given the answer? Okay. Check, everyone, everyone. I'll give the answer just like that, okay? It's very simple. Okay, take time, not a problem. Hmm. Others, you know what you people are doing? You are thinking too depth. When you think too depth, trust me, you will make it wrong. The moment you think too depth, you make it super wrong. Make it simple. The best way of answering question is making it simple. That's the brilliant way. Oh my God, oh my, oh my God. Okay, Ritika, everyone, Sisira, all the students answer my question. Padmavati, why two answers are coming? Okay, four, you are saying A only. Okay. You know, I will tell you the one simple difference between a person who is calm-minded and the person who always get excited. The person who always get excited will choose every single option wrong because he get confused. He always get confused with options which he already know. Every student answer my question. In case of a eukaryotic organism, 5' capping occur in which area? All the students answered. In case of a eukaryotic organism, 5' capping occur in which area, which location? Tell me, Padmati, tell me, Archana, where the 5' capping is occurring? Everyone answer. Where it occur? Nucleus. No one is seeing the option D, right? Huh? Capping of eukaryotic mRNA occurs exclusively in the nucleus of the cell. Now tell me, is this statement is a true statement or a false statement? This statement is a true or a false? Okay, this statement is a true statement. Okay, you got that point correctly. Okay, option A says that RNA editing can occur via the deamination of the cytosine residues, leading to the formation of uracil and thus change in the coding sequence. Now tell me, Archana, is this statement is a true statement or a wrong statement? Option A, Ritika. Tell me, Nitya, you are also there, right? Okay. Wait. Okay, look into this. Just wait. Let me open it. You'll automatically understand. One second. No, I want to show that option to you people. Processing. Okay. All the students, please look into this question correctly. Look into the point. Okay. This is the option, right? So look into this. What is the question option they have given in the exam? What is that? RNA editing occurs via deamination of the cytosine, leading to the formation of the uracil. That is the option one. Where is this here? Yeah. So deamination of the cytosine. All the students look into this picture. What the deamination of the cytosine is going to give rise to. Everyone look into this. What is this? All the students answered. Of course, anyone else, others? Is it a uracil or is it a uridine? My question to all of you, is it a uracil or is it a uridine? First, it is converting into what? Very good. First, it convert into the uracil. Later, uracil convert into the uridine. No need to get, no, Archana. First, it is uracil. Then, it will convert into the uridine. And the point is that whether it is a uracil or uridine, both are different sequences which cannot found in the DNA. You're getting my point. So now listen, option B. Every student answer option B first. The major spliceosomal complex mediate the removal of the group 2 introns. Every student answer my question. We already studied, right? Group 1 and group 2. They mediate what kind of a splicing? We studied or not? Group 2 and group 1 will mediate what kind of splicing? Self-splicing. But they have given the option that they mediate the spliceosomal mediated splicing. Correct, na? So this will be the option B will be the wrong statement. You're getting my point. Option A will be the right statement. Now option C says that trans-splicing seen in the trypanosomes allow the formation of a multiple gene products. Now you tell me, friends, are we going to get the multiple proteins by the trans-splicing? Are we getting the multiple gene products by alternative splicing? All the students has to answer. Are we are getting the multiple gene products by alternative or the trans-splicing? So option C is the wrong option. Here also they have given the answer as a D. But remember, friends, capping of eukaryotic mRNA, I am talking about eukaryotes. It is exclusively in the nucleus only. But most of the people challenged this particular question because A is the correct answer, D is also the correct answer. But in the given answer key, they have given only option as the D. Okay, but this question got challenged, but we don't know what may happen in the final. Okay, answer A is correct, D is also correct. B and C are absolutely wrong statement. Clear, everyone? No need to get confused with that. Slowly analyze this. You can get it correctly. Correct, na? See here. But the point here, this statement is a true statement. Capping of eukaryotic mRNAs occurs exclusively in the nucleus of the cell. Most of the eukaryotes. But the point is that they did not mention, friends, whether it is a higher eukaryote, whether it is a lower eukaryote. If you take an example of a lower eukaryotics like what we can say, trypanosomes, it may also occur in the cytoplasm. So that's why this statement is also contradictory statement. In this particular condition, if you choose option A and option D, anyone will become the right. In general, will be the nucleus. Okay. So any option you can choose here. So the last question here, question number 72. Think it slowly and answer the question. I'll give you this, don't worry. Yeah, take your time and answer. Actually, there is a DNA repair mechanism question also there, but I will explain that question once we complete the genetics chapter. Take your time, think it. They're asking mutant DNA polymerase, mutant. Take your time. What about others? Everyone give the answers. See, friends, now tell me. A mutant DNA polymerase found to have a higher error prone as a result of mutation, it is going to synthesize only short DNA fragments. In the statements below, potential explanations are given. Easily you can choose the answer, right? Very easily you can choose the answer because there is a no proofreading in the 5' to 3' direction. Option A is wrong. Eliminate option A, eliminate option C. You have only two options. Proofreading is what? Proofreading only in case of a 3' to 5'. So this get compromised because there is a mutation. Now this is the DNA. This is the polymerase enzyme. When the polymerase enzyme undergone the mutation, polymerase enzyme attaches to the DNA and keep on falling down, keep on falling down, keep on falling down. So this is right. Polymerase tend to frequently dissociate. C is also right. Option D. So undoubtedly you can choose the option here itself because B is undoubtedly true statement. The polymerase is unable to unwind. First of all, why the polymerase is involved in the unwinding, friends? Helicase is involved in the unwinding. What is the answer? Option B and C. That is the right option. Is it clear, everyone? Nothing like that here. Everything is easy. Okay. Everything is depends on our intelligence. Okay. But the point is that I don't encourage any one of you to answer every single question from the unit 3, every single question from unit 2, every single question from unit 4. If you do that, that is the most stupidest way of answering the question. Out of 7 questions, our goal is to answer only 3 questions because we have not dependent on all the in single unit, right? We are reading 11 units. If you answer 1 unit 2 part C question, 11 unit, you will answer 22 questions. So undoubtedly you will crack the exam, no doubt about it. Okay, but again you require a lot of patience. So in the afternoon shift, what are the question number? Question number 6, 12, 26. So these 3 which are 6 marks. Then in the part B, sorry, part C, 28, 30, 33, 47, 72, because it's a jumbling, right? So the question number will be varied. So 5 into 4, 20, 20 plus 6, 26. Morning shift also we got 26 marks. Okay. And 20 plus 6, 26 in the morning shift also and in the evening shift also. Because Nitya, we have to practice more number of questions. Then only we can answer at least a few number of questions in the final exam. So remember, in the morning shift and evening shift, we have a equal weightage. And friends, what is common in the morning and evening shift? Listen to me correctly. In the morning shift also, we got the question from translation. Evening shift also, we got the question from translation. Here we got only 1 question. Here we got 2 questions. And here it is a part C question. Here also it is a part C question. Post-transcriptional, we got part B question in the morning shift. Evening shift, we got the part C question. Replication, in case of a morning shift and evening shift, both we got the 4 marker questions only. So that's why what are the crucial topics? Replication, especially transcription in eukaryotes, post-transcriptional processes, and translation. And what is common in the morning and evening shift? Post-transcription and operon. Operon is the most common topic in the morning and evening, right? There is another common topic I will explain you in the genetics, that is called bacteriophage. That is also most commonly asked. So an average, friends, 26 to 30 marks were appearing in the unit 3 in both the morning shift as well as the evening shift also. Now let me complete the plant physiology, then we will solve the plant physiology as well. Plant physiology will contains more marks than this, more tougher than this, more logical than this, but more important than this. Okay, in the tomorrow morning class, we will starting the biggest unit, that is the unit 4. I will solve the questions then and there itself in a very different way. Okay, by that also becomes more easy for us. Okay, perfect. Then I'll share this lecture also. I'll see in the tomorrow morning class. Plant physiology will take another 5 to 6 classes because solute transport also very big concept. It will take 3 classes for me to finish that. So we'll take 5 to 6 classes to finish the plant physiology. After that, immediately we'll solve the cell biology. We'll start cell biology, and tomorrow morning onwards, we'll start the unit 4. Okay. Okay, friends. That's all for now. I'll see you in the tomorrow morning class. I'll share these question papers to all of you. Okay. Thank you all. Thank you. I hope the discussion is useful to all of you. Thank you.